Well, welcome everyone so far, and welcome to those who will get to be here. I'm Howie Rosen. I'm a neurologist at UCSF and part of the clinical steering committee that is organizing these series of webinars. Very happy to see many people interested in this. Without further ado, I think, Hannah, should we start the first one?
Sounds great.
Okay, so before I do that, I'll just say that as we're doing this, we have a pretty tight timeline, and so we'll have very little time for questions, but I'm encouraging people to go ahead and put comments and questions in the chat. I'll try to monitor them, and if we have time after each talk, we'll pick one or two that the speakers can address. So looking forward to the talks. The first one is from Julio Rojas from UCSF. Take it away, Julio.
Thank you, Howie, and thank you all for having me today. I'll share things that are related to fluid biomarkers, blood biomarkers in Alzheimer's disease and other neurodegenerative disorders that are close to have an impact on clinical work. And I condensed it, since we have only 10 minutes, I condensed it in 5 main points. So the first point is there's a lot of value in blood biomarkers. The main thing is that they are deployable, they are convenient, they're accessible, potentially inexpensive. And we can talk today about these biomarkers because of the great progress we've made in technology. The evolution of antibody-based detection technology, which is what predominates, what is touching the clinic, started in the 70s with this immunoelectrophoresis. The sensitivity was very low. This allowed maybe to detect albumin and antibodies. But then in the 90s, ELISA predominated. The sensitivity improved a lot to the nanogram range. And in this century, we've seen third generation methods, specifically electrochemiluminescence and single molecule arrays that greatly increase sensitivity. And this is really combined with the better understanding of biology that we have, that we're able to deploy this type of biomarkers. Single molecule arrays are bead based, and so there's a bead that is coated with antibodies. I used to think, or I like to think of it as a ELISA on steroids. However, there are other methods that are revolutionizing the field. One of them is an automated panel that could deal with the problem of having different standards and results by lab. So a technology like this could make results comparable. Then there are other bead-based technologies that could allow, for example, multi-analyte profiling. This technology, for example, can allow you to screen for 500 analytes at the same time. Very efficient and still very sensitive. Another bead-based assay, immunomagnetic reduction. There's great data on detection of amyloid, beta 40, 42, p-tau 181, and separating, great discrimination between groups. And then immunoprecipitation linked to mass spectrometry. Here is the separation of the analysis also based on a bead coated with antibodies, but then is a link to mass spectrometry, which makes the method really, really specific. So in the last 10 years, we really went from not having an assay in blood to a revolution. So the first Simoa publication came in 2010, and then a number of blood assays for the relevant biomarkers that have been useful in CSF for Alzheimer's disease started to emerge. Then NFL came in, which has been a great marker of axonal degeneration, a non-specific marker. And then some assays not only started emerging, but started testing the clinical value for diagnostic and prognostic purposes. We have the fully automated blood panel for some markers in 2019. And then we have the emergence of p-tau 217 and p-tau 231 this year, which are also very good markers of diagnostic markers. So great scenario so far. The second point, the biomarkers have high potential for clinical value, and I'll just dwell a little bit into the data here. So with immunoprecipitation and mass spectrometry, there's data showing that The ratio of plasma abeta 42 to 40 is lower in males and ApoE carriers. As you can see here, this is a graph showing age versus levels. And there's a clear difference by sex, clear effect of the ApoE genotype. And also in this study, which is a study of 158 participants who are cognitively normal, this assay in blood was able to discriminate people with a positive amyloid PET versus those with a negative one. And the accuracy was really spectacular for what we've seen in assays in blood. Also, in a prospective follow-up of these participants, the plasma assay was able to predict who will convert in the next few years over, a follow-up of 8 years from negative to positive amyloid PET. So if you had an elevated or an abnormal test here, the risk increased 15-fold to developing a positive amyloid test. So this is a great tool. Is with IPMS, again, mass spectrometry, not very accessible in a lot of centers. So there are other studies that have shown the value of their biomarkers. For example, this study in which we participated, that you look at p-tau 181 with electrochemiluminescence, very good performance at discriminating different type of different subgroups. This was a study that included around 362 participants that included AD, FTD, and healthy controls. And you can see here the sensitivities and specificities for discriminating the different groups by amyloid PET, tau PET, amyloid PET, only in controls, just similar to what we saw with amyloid beta ratios. And in MCI, that was the highest area under the curve, and then even higher for tau PET in MCI only. So it is a great discriminator in the Alzheimer's realm. Also, not too shabby in differentiating path-confirmed Alzheimer's versus path-confirmed FTLD. So this was a great tool, and it's still happening or still improving. Another phosphorylated form of p-tau 217 was tested in another big study that included 3 cohorts. So 1,400 participants. The first cohort tested plasma p-tau and differentiated neuropath, confirmed AD from non-AD and neurodegenerative diseases with an area of under curve of 0.89. And the p-tau 217 concentration strongly correlated with tau tangles in patients with beta amyloid plaques. In this figure, you're seeing a second cohort that differentiated clinically diagnosed AD dementia from other neurodegenerative diseases. And you can see here how p-tau 217 compares to other biomarkers, and is at least as good or close to CSF p-tau and tau PET. And then another cohort here tested asymptomatic carriers of AD causing mutations, and p-tau was elevated and predicted the carriership. So this is a great tool for screening, specifically in those conditions where it could be rare, like a gene carrier. Great screening tool. Another piece of information that we know in recent years is that plasma tau, in contrast to phosphorylated tau, is not a great biomarker for amyloid status in dementia. Here you can see it in different ways. You can split it by severity of cognitive impairment. And you can see here amyloid positive versus amyloid negative. There's really no difference. A big contrast to what we see with phosphorylated p-tau. There are assay-related issues about this. I'm happy to talk about it more, but we really cannot discriminate a lot with plasma tau, at least with what is commercially available. However, if you use specific antibodies that target specific regions of tau, we have data showing, for example, that antibodies that are directed against the N-terminal tau are actually good at discriminating people who are amyloid positive or negative, and there are correlations with progression and atrophy. So it has to do probably with an analytic aspect. So plasma tau may not be out of the picture. We just need to pay attention to where in the tau protein it's binding to. Okay, number 3, neurofilament is potentially valuable, especially for clinical trials of neurodegenerative diseases. An exciting study that we conducted was this study in two big cohorts of carriers of FTLE causing mutations. And one of them was the North American version, all FTD, and then the European version. These were large studies of about 290 patients each. And it included asymptomatic carriers, and then carriers that had symptoms, either a prodromal or full phenotype. And elevations of NFL predicted those asymptomatic carriers that eventually started to become symptomatic. So this is a great tool for screening and for selecting who in a clinical trial might benefit from a potential drug whenever they are available, as opposed to enrolling people and having the uncertainty on whether those people within the duration of the trial will convert. This is a great screening to be able to select those patients. Another reason why neurofilament is so important and is because based on the work that we have or data emerging from the MS research and multiple sclerosis, upon introduction of treatment, blood neurofilament levels tend to decrease. And these are different cohorts at different time points here on the x-axis and different MS therapies. And you can see that in all of them, the tendency is to normalization towards healthy control levels. So this could be a great tool. And we don't really have something for neurodegenerative diseases, but some data started emerging, including some work presented this week in the American Academy of Neurology meeting, showing that these some effects in blood are seen upon instauration of treatment for certain conditions. The best example that we have published or we've seen published is in CSF, but now we're starting to see emergence of data in blood. This is a study in which they treated with nusinersen for spinal muscular atrophy. So this is exciting data. Now, fourth point, we need to embrace new paradigms for discovery and validation. And this includes paying attention to cohort-related factors, asset-related factors, analytical factors. And this includes having a really clear pipeline on replication in different cohorts, using the right comparisons. For example, always including a control group, always running all the assets in the same batch, pre-specifying the analysis that we'll run, and then having this idea that before introducing it in clinical practice, there's got to be a validation, and preferentially global validation. And which brings me to this other aspect. We actually need to embrace the need to address this issue of diversity and equity inclusion in blood biomarker discovery. There is a need to address the differences that we know exist in dementia at the racial, ethnic level. There are reports showing that socioeconomic status may determine the biomarker status, at least from what we know from CSF and amyloid PET. But we really need to address the problem that sometimes there's bias in because of the differences in expression of biomarkers in different ethnic groups or racial groups, or even between gender. This could limit some access to participation. For example, in this study in the lower corner, left lower corner, the differences limited access to Hispanic, non-Hispanic Black participants to the trial because they didn't meet the criteria, when we know that that population is actually affected. And then recent studies showing, for example, that there are indeed biological differences determined by genetic variants in levels of TREM2, a factor that mediates inflammation and may modulate the risk of having Alzheimer's disease. So we need to embrace this as a field, and a lot of the research should start considering these factors. Okay, I'm gonna stop there. Thank you.
You're a pro, Julio. Nice job. That was great. I didn't see a lot of questions. A couple of nice comments in the chat. Somebody asked about seeing one of Julio's slides again. One of the ways I might respond to that, Hannah, do we have a place where we're archiving these that people can download them or access them afterwards?
This talk will be recorded and on YouTube, uploaded to the NAC channel, so you can always reference it back there. And if people would like to have me link their presentations in a follow-up email, I'm more than happy to do that, but I'll leave that up to the panelists' discretion.
Okay, so somebody can write Julio if they like, and also that so that was Shania Jiang's question. I may be mispronouncing that. There's another way to see the talk again.
Yep.
Thank you, guys. The next speaker is Lenora Higginbotham from Emory University, and looking forward to hearing her talk.
Okay, here we go. Okay,
Perfect.
thank you. Thanks for having me. Like I said, my name is Lenora Higginbotham. I'm an assistant professor of neurology over at Emory University Brain Health Center. I'm also a member of a multidisciplinary proteomics lab at Emory, run mainly by Drs. Alan Levy and Dr. Nicholas C. Fried. And I was asked today to talk a little bit about proteomics and Alzheimer's disease, but I narrowed it just a little bit to talk about mainly network-based strategies or systems-based strategies to biomarker discovery, proteomic biomarker discovery in Alzheimer's disease, mainly because it's a little bit of what our lab specializes in. We use primarily an unbiased systems-based approach. I'm going to talk a little bit about that in a second, but I just want to talk about a little bit about the rationale for this work. So as we all know, increasing evidence indicates that AD pathophysiology extends well beyond hallmark amyloid and tau accumulation. There's actually evidence for a decades-long cellular phase of Alzheimer's disease, which is comprised of not only just functional neuronal mechanisms, but also glial and endothelial mechanisms as well, and that this contributes significantly to irreversible damage. And core AD biomarkers currently, which focus primarily on abeta and tau detection, really poorly reflect this intricate, really diverse cell-mediated pathophysiology, and suggest a need for systems-based approaches, network-based approaches. I wanted to highlight this paper, this great paper by DeStrooper and Karen, published in 2016, which really outlines a lot of the evidence for a cellular phase of Alzheimer's disease, really talking about moving away from the amyloid hypothesis, as many have been doing over the past several years, which posits this really neuron-centric linear cascade where everything is initiated and then sustained by a beta accumulation. And really incorporating this cellular phase in which, it may be triggered by a beta accumulation, amyloid accumulation, but really can be described as a more self-sustaining detrimental part of Alzheimer's disease, involving these feedback and feed-forward mechanisms with astrocytes, microglia, vasculature, all these different cell types. And that if we're going to develop appropriate biomarkers, effective biomarkers, and really move into precision medicine, the field must incorporate this holistic view. And this is another figure from that same paper, which really lays out kind of the model for this cellular phase. Having Alzheimer's disease first start off with a biochemical phase years before clinical symptoms, in which abnormal A beta and tau accumulate, triggering a cellular phase, which at first is homeostatic, but after a while turns into these detrimental self-sustaining feedback feed-forward loops involving neurons, astroglia, microglia, all these different cell types, and then eventually causing chronic inflammation, circuitry imbalances, leading to cognitive symptoms, and then ultimately Alzheimer's disease. And so here's where systems-based proteomics comes in, and I like this quote from this neuroproteomics book, which talks about the evolving relevance of neuroproteomics in Alzheimer's disease. Really talking about how systems-based biology strategies can really provide a full depiction of the complete molecular pathogenesis of all forms of AD. And as I mentioned before, as we recognize more and more the individual, racial, all these different kind of nuances and how people, the biology of the disease for various different people, how we can move into this really detailed, nuanced, like biomarker detection and customize and tailor our medicine. So when I entered proteomics a long time ago as a med student, we were using some network analysis, but I describe it as mainly biased. Started in 2011 with like doing 18 cases at a time. But we would, start off with our control and dementia. And since this is a biofluids talk, let's say you have CSF or plasma. Our proteomics are mainly mass spec based, so we would do our mass spec and then our classic differential analysis, produce our volcano plot and our hundreds of proteins or whatever that were significantly altered in disease. And then out of those differentially expressed proteins, we would do a network analysis looking for functional links between proteins, mainly based on previously described or known relationships between proteins in the literature, and then replicate and do target prioritization. However, we kind of eventually moved a little bit away from this, recognizing that as we learn more and more about the utility of networks and systems-based approaches, mainly from transcriptomic studies, that this classic approach, sort of a biased approach, does really little to better define global brain-based pathophysiology or really contextualize biofluid markers within this complex biochemical and cellular phase of disease. And so that is kind of what triggered our lab, at least, and several collaborators to move into several years ago, this kind of unbiased co-expression network analysis. A lot of this type of network analysis was adopted from methods from transcriptomics. But the idea is to really start with the brain and really define what is going on with all these cell-mediated techniques and look at proteins from this global scale. And the way that we do this, we use a program, at least mostly we use a program called WGCNA. It's an algorithm, but there's other co-expression network analysis where it takes the entire brain proteome, the thousands of proteins, looks at their individual expression levels, each of those proteins across individual cases, and then we'll actually organize these proteins into groups or modules of proteins based solely on their expression levels across cases. So proteins that have very similar expression levels across various cases will be grouped together and form modules. And this has given us a really really kind of complete and interesting overview, proteomic overview of the AD brain, which I'll get into in just a second, and really has identified not just individual biomarkers, but groups of markers that are really relevant to disease. And then what we're trying to do now recently, within the last couple of years, mainly, is integrate what we've learned from the AD brain proteome into our proteomic analysis of biofluids and really contextualize the biofluid markers that we're getting in this overall global picture and develop multiplex proteomic assays. And this is just a visualization of what co-expression network analysis is doing. It's taking all these proteins and in unbiased fashion based on co-expression, grouping them into modules or groups. The nice thing is even though this is largely data-driven on co-expression, these modules often have a lot of biological relevance. And so when you look at gene ontology analysis of these modules or do other even genetic analysis, genetic marker analysis of these modules will map to very specific cell types, organelles, molecular functions very, very consistently. So not only is it data-driven, but also biologically relevant. And like I said, we've really been doing this over the last several years, and we've learned a lot. This was one of our first major papers looking at this multi-network approach, and we've been doing this in Alzheimer's disease, asymptomatic Alzheimer's disease, so brains with amyloid pathology but no symptoms, and then controls as well are our main groups. And when you produce these networks, this is what they look like. I won't go into it in detail, but there's this hierarchical clustering method that's used in WGCNA. And then when you get your modules, they have different colors, different numbers. And what you can do is correlation analyses. You can look at how well their levels across Alzheimer's disease and controls, how well they correlate with amyloid, tau, pretty much whatever you want, if they're enriched in genetic risk factors, whatever. And then also you can take the weighted average of their levels across whole groups, and then look at each module and its general expression pattern across whole groups of disease. So in this case, like if you look at astrocytes and microglia enriched modules, you can see that they're increasing gradually across disease here, mainly saying the same between control and ASM-AD and increasing in AD here. And then oftentimes the central hub proteins, we can identify those as well. These are the ones with the greatest connection to that module, the greatest basically I'm getting the wrong word, but basically their expression levels across cases are really exemplary of the module. And then we've looked at probably at least hundreds, if not thousands of brains now over several years, including not ending. We're doing another one with a different type of mass spec, but this consensus analysis, because what we were seeing is that didn't matter what we built the brain network proteome on, if we built it on one 80 cohort versus another 80 cohort, or if we built it using one type of mass spec versus another, we see a lot of consistency between various modules that come out and various modules that change within disease and various hub proteins. So down to the hub protein level, we get a lot of consistency. Basically what this has really led to is, this is the proteomic basis for the AMP-AD consortium, which has really taken this national group of Alzheimer's disease research centers and really it's designed to get back to systems-based or really designed to get to systems-based, network-based strategies, defining brain-based pathophysiology to really yield new targets, new biomarkers for Alzheimer's disease. And so just to summarize a little bit before I briefly talk about how we've gone into the CSF is that, these disease-associated modules, their hub proteins are highly preserved across cohorts. We see specific cell types driving the organization. We can do GWAS target enrichment, and so genetic enrichment, and look at causative roles, potential causative roles for the modules, and several of the modules increase in ASM-AD. And these are just some of the things we've seen: inflammatory myelination, RNA binding, increasing in AD, synaptic mitochondrial cytoskeletal, and very common hub proteins. And so since this is a talk about fluid-based biomarkers, I just want to make sure I get to this integrated strategy that we're doing, which is based on primarily in a paper that we published last year. And what we did is we took one of these characteristic brain networks built on 40 Alzheimer's disease and other neurodegenerative disease tissues and controls. And then we took CSF proteomic analysis and did a classic differential expression analysis. And what we wanted to see from these two separate proteomic analyses is where the key points of overlap were. So we did a Fisher's exact test, looked at the different modules, like you see here, there were 44 in this group, and then looked at which modules really significantly overlapped with the total CSF proteome, or just upgoing or downgoing CSF proteins that we called potential biomarkers. And we found that out of these 44 brain modules, we really just needed to focus on about 15. They were the key points of overlap or overrepresentation in the CSF and its associated biomarkers. And then when we were able to break this down by gene ontology, et cetera, we found that these 15 modules really did, and their associated CSF biomarkers, represent a very diverse reflection of cells and pathophysiological mechanisms in disease. We then did a lot of replication over hundreds of CSF samples to get these 200 and so CSF biomarkers down to 60 highly reproducible biomarkers. And we came up with these panels, which actually were nice because they had some sort of directionality in disease that you could see. This is kind of like an average expression of all these proteins together. You can see they're increasing in disease and several and decreasing here. And we did this over hundreds and hundreds of cases, over 500 cases, and found a lot of replication there, and even a little bit of disease specificity down here. And then finally, in my last couple of slides, I just want to show how stuff like this may be helpful in teasing out nuances in patient populations. So we looked at these panels in Alzheimer's, asymptomatic Alzheimer's disease, and we found that there were two different groups actually when we looked at our top changing proteins in these panels, and that you had an ASM-AD group that was more AD-like and one that was more control-like. And this suggests that maybe we can risk stratify patients in early disease. And we've actually also applied these marker panels to a phase 2 clinical trial that is not yet to be published. So I'm not going to go into the results of the clinical trial, But we used atomoxetine versus placebo, and atomoxetine is known in mouse models to reduce amyloid pathology. And we also saw that in the patients that participated in this trial and got treated with atomoxetine, that while the glial and vascular panels weren't affected very much, the neuronal panels were actually significantly altered. And this can be used one way as just confirming target engagement or what cell types different drugs are targeting. And so in conclusion, this unbiased network proteomic approach could be very useful. We are currently actually validating a lot of our, some of those new biomarkers that I mentioned with targeted mass spec. And we have, high hopes for a lot of this work, and just that it will help with our nuanced detection of disease and profiling. And that's our lab. Thank you.
Thank you so much, Lenora. It's amazing how much you can learn in 15 minutes, huh? So that was great. I think I want to keep going, but I think we'll have some time for questions at the end. I think you should save up questions for all the panelists, and I'm encouraging people to put the questions in the Q&A section. We'll also monitor the chat, but if you have questions, try to put some in, and hopefully we'll be able to get to them for a few minutes at the end. It can be for any speaker. So without further ado, I'll introduce Adam Brickman from Columbia University.
Thanks, Alan. Thanks, everyone, for the invitation. I'm having some slide issues here. I'm going to reshare my screen, if that's okay. Okay.
It looked good to us, just so you know what you had.
Thank you. Yeah, I think I shared the wrong one.
Okay.
Okay, can everyone see that?
Yes.
Okay, great. So thanks so much for the invitation. My name is Adam Brickman. I'm a neuropsychologist at Columbia University, and today I'm going to be talking about community-based assessment of fluid biomarkers in dementia. The assigned title had the word barriers and opportunities in it. I think it's all opportunity, to be honest. These are my disclosures. And when we think about biomarkers, we think about disease-specific biomarkers, and they can be used to detect the presence of disease, to stage the disease, to predict treatment response, to test drug efficacy, monitor compliance, and monitor disease progression. I'd say that we often forget, however, that the incorporation of biomarkers can also help us identify relevant biological changes that might contribute to the clinical presentation or pathogenesis of the disease. And this concept is particularly relevant in Alzheimer's disease and related disorders, where I would say it's still the case that the exact mechanism and pathological features are still incompletely understood. So of course, the field of AD research has been revolutionized by the development of neuroimaging and CSF biomarker approaches that can be used to characterize the amyloid, tau, and neurodegenerative changes that are hypothesized to capture the primary pathophysiological changes of the disease. This shouldn't be news to this audience, but on the imaging side, amyloid and tau PET scans, such as those acquired with florbetaben or MK6240, which we use a lot at Columbia, and structural MRI scans are often used to capture the ATN cascade. Similarly, on the CSF side, ELISA-based assays can be applied that can characterize abeta pathology, p-tau, and total tau, and NFL, and other related biomarkers, as we've heard so far. Despite the accuracy of imaging and CSF biomarkers for AD, there are limitations, of course. So first, institutions, and even in some cases, entire countries, don't have access to radiopharmaceuticals. They're often expensive. Patient and research participants are often resistant to the procedures or the risks. And there may be contraindications, and these issues, of course, lead to selection bias in research studies, and in some cases, limited access in clinical settings. So as we've heard already, there's really been an explosion in blood-based biomarker development, and this has really been ushered in by advances in ultra-sensitive assays, some really motivated scientists, and this is just a sampling of some of the papers that have come out in the past few years that highlight the accuracy of some of these blood-based biomarkers that have come on board. So there are obvious potential advantages to blood-based biomarkers for AD and related disorders. Blood draws are considered, I think, by most adults to be routine. So the barriers related to discomfort or distrust are reduced. No specialized equipment is necessary really for blood draws in most clinical settings, although of course specialized equipment or analytic platforms are necessary for the analysis. And these advantages of incorporating blood-based biomarkers lead to less bias in research studies and greater overall access. I'm not saying that there isn't still a role, of course, for CSF and imaging-based biomarkers, but blood-based biomarkers certainly opens the door to a lot more inclusivity. And now that the question is really, do these blood-based biomarkers work in all settings that we want them to work in, or do they work well enough so that the pros still significantly outweigh the cons of incorporating them? So this is a slide that I stole from my friend and colleague, Maria Gleamore, and I think it really captures a lot of how I think about some of these things. So different study designs are really best suited for different scientific goals. And to test causality, for example, we would incorporate or use true experimental designs or clinical trials. To maximize statistical power and measurement quality, we rely on volunteer cohorts and clinic-based studies. And to date, the implementation and the development and the testing of blood-based biomarkers have really been restricted, probably appropriately, to these types of designs. However, to test the generalizability and the applicability across diverse populations, the strongest designs are really ones that incorporate population representative cohorts. And so we started incorporating plasma-based biomarkers into our community-based studies at Columbia, and I'd like to share some of our preliminary findings with you today. These are based on a paper that just came out, I guess about a month ago, in Alzheimer's and Dementia. And really the goal of the paper, the goal of the preliminary analyses were to examine the association of blood-based biomarker concentration with a pathological AD diagnosis, with cross-sectional clinical AD diagnosis, and with future clinical AD diagnosis. And these are all community-based participants that came from the Washington Heights Inwood Columbia Aging Project, or YCAP, which is a multi-ethnic and multiracial community-based cohort that represents the community or the population surrounding Columbia University Medical Center in Upper Manhattan. And so these are really pilot data or preliminary data that we used in our initial analyses to motivate a larger scale study, which looks like hopefully it'll get funded. We have some preliminary positive news about that. So YCAP is a prospective community-based study. It was started by Dr. Richard Mayu, I think informally in 1989, formally in 1992. It's gone through many versions, and the current leadership includes still Dr. Mayu along with Dr. Jennifer Manley and myself. Over the years, we've enrolled about 6,500 older adults. These are Medicare eligible residents that were recruited with epidemiological approaches. So we didn't put up flyers, we didn't have clinic referrals. We used methods that sampled proactively community-based members that represent the community around the medical center. And this yields a sample that's approximately equal numbers of non-Hispanic white participants, non-Hispanic Black or African American participants, and Hispanic or Latinx participants. And those are the three main racial and ethnic groups that comprise the community around the medical center. So let's talk about the neuropathological diagnosis. For these analyses, we selected 113 YCAP participants who had come to autopsy and who had stored plasma samples, which were acquired about 3 years before death. They were characterized neuropathologically according to NIA criteria and the Alzheimer's Association guidelines. And we grouped participants as having high degrees of AD neuropathological change, compared with those with lower degrees of neuropathological changes. And this approach has been used in a lot of different studies that have looked at fluid biomarkers. So we used MSD and Simoa-based platforms to quantify concentrations of plasma p-tau, abeta, total tau, and neurofilament light chain biomarkers in this study. So this is a bit of a busy slide. Let me walk you through. This is comparing controls at autopsy, the lower AD groups with the high AD neuropathological change group. So age at time of death and race ethnicity were similar in the two groups. As expected, AD patients were more likely to have E4 allele. When we compare absolute concentration values in life, we see that the patient group had higher levels of p-tau 181 and 217, but were similar on the other AD biomarkers. When we constructed ROC curves, you see that both p-tau concentrations were able to classify patients accurately. Although statistically similar, p-tau 217 was numerically better than p-tau 181, and this was the case in all the analyses that I'll show you. 217 outperformed numerically, but not statistically 181. NFL also helped discriminate the groups, and when all markers were considered together, the area under the curve was about 0.9, and this value was similar if we considered each p-tau marker separately. So if you examine the 4 levels of AD neuropathological change, what we find is that the A-beta 42/40 ratio concentrations were numerically or slightly lower in the AD group, but the effect sizes were really quite small. On the other hand, p-tau 217, and not shown here, but the exact same effect, p-tau 181 did differ across the patient groups in a dose, in a monotonic or dose-dependent fashion. So what happens when we look cross-sectionally at the clinical AD data? We selected 300 participants from the YCAP cohort population, and our intention was to include 100 from each of the racial and ethnic groups that represented the study. We analyzed the plasma samples taken at the time of a single diagnostic visit, and the diagnosis of AD was made by a consensus panel that reviewed the neuropsychological, the historical, the medical data, but not any biomarker or neuroimaging data. I think I said all that. And here's another busy slide, but let me walk you through. This is comparing the clinical AD patients and the non-AD controls. The AD patients were older, but had similar sex, gender frequencies, ApoE4, and race ethnicity distribution. Bivariate comparisons of the biomarker levels showed that, again, p-tau 181 and 217 were reliably elevated in the patient group. And here the NFL concentrations were numerically higher in the AD group, but there's really no difference in the amyloid or the total tau concentrations between the group. When we look at the ROC curves, the first thing to note is that they aren't the mountainous ROC curves that we typically like to see in these types of classification studies, and that there's a lot to think about there. But statistically, p-tau 217 and 181 were really the best discriminators. Again, with 217 being a little bit higher, and here NFL contributed a little bit. When we look at all the biomarkers combined and classifying clinically defined AD participants from controls, the AUC is really only about 0.67, which I think is a bit modest. So it's important to note that although preliminary, we didn't see differences, as I said before, in the level or the concentrations, maybe I didn't mention it, of the AD biomarkers across the race and ethnic groups, nor did we see differences in the patterns across race and ethnicity groups included in the study. With larger numbers, we're going to be able to examine stratified models and the relevant moderators and mediators across the different racial and ethnic groups. But in these preliminary findings, we didn't see much difference. So let's consider sort of incidental diagnosis or incident diagnosis of the control participants at the time of blood draw. 71 of them went on to develop AD by their last available clinical follow-up visit, which was about 4 years after the blood draw on average. And we ran a survival analysis to examine the odds ratios. And here we used a median split of A-beta ratios and p-tau concentrations. And again, another busy slide, but let me summarize for you. The results of the survival analysis revealed that, again, p-tau concentrations, and in this case, A-beta levels and NFL concentrations predicted future AD diagnosis. Here I'm again displaying the results of p-tau 217, but the effects were really quite similar with 181. Again, with p-tau being the strongest predictor of future diagnosis, and in the higher median group, more than doubling the risk of clinical AD over the follow-up period. So what's driving our diagnosis? So if we were to assume, just for argument, that the plasma p-tau 217 is which is our best performing biomarker, is 100% accurate. Some people in the cohort have high p-tau levels but are diagnosed as cognitively unimpaired. Others have low p-tau levels but are considered to have dementia. We can calculate the Uden index, which is the cut point based on sensitivity and specificity that best discriminates the group. And here I'm showing that cut point in our data is 0.34 against the neuropathological diagnosis. So if we apply this cut point to the 300 participants in our clinical sample, you can see that the dementia negative p-tau most of the dementia negative people are also p-tau negative, but somewhat surprisingly, most of the dementia positive folks are p-tau negative as well, based on this cut point. So we looked across these 4 different groups at cortical thickness in AD signature regions as a marker of neurodegeneration. And although the p-tau positive cognitively unimpaired participants had numerically lower cortical thickness compared with the p-tau negative clinically unimpaired group, both groups that were diagnosed with dementia had statistically reliable cortical thickness, lower cortical thickness in AD signature regions relative to the p-tau negative clinical negative group. And these data, I think, suggest that neurodegenerative changes, irrespective of tau status, are driving a dementia diagnosis in a community-based study. Of course, this conclusion depends a lot on how much the plasma marker is really capturing what's happening in the brain, and we'll play this out once we have literally thousands of people with both imaging and biomarker levels. So, our pilot data suggests that p-tau concentrations outperform other plasma biomarkers in the community. Plasma biomarkers are more strongly tied with pathological diagnosis. P-tau concentrations together with A-beta predict future clinical AD diagnosis, but A-beta 42 ratio alone isn't particularly effective cross-sectionally or with respect to autopsy data. And we didn't really find evidence in these preliminary data of race ethnicity differences in biomarker concentrations or their associations. So just a word on race and ethnicity differences in biomarkers. I just want to emphasize that diversity doesn't equate to representation in the studies. You can have a diverse study cohort, but you really need to consider how sampling strategies and differential participation. So when we look at a lot of the published papers, some of them were displayed in an earlier talk, we really need to ask if the sampling strategies or differential participation might be leading to conclusions about differences across race and ethnic groups. In some studies, yeah.
You think it's okay if we wrap it up?
Sure, I'll wrap it up with this.
Give Camille his minutes. Go ahead.
My apologies. I thought I had timed out right, but I'll leave it with this thought that in some studies that use genetic ancestry, those two can also be driving racially patterned social forces that lead to observed differences. So with that, I just want to acknowledge my collaborators on this and thank you all for the attention and apologize for going over.
No apology. Fantastic talk. Lots to think about. Thank you, Adam. And obviously, time is not as luxurious as I had hoped. Well, we're going to go right on to Camille. But meanwhile, I'll point out, panelists, there are a couple of questions in the chat and a couple in the Q&A, and I think we should just go ahead and answer them by text. And of course, you could all be contacted individually for additional questioning. So without further ado, Camille Borkowski from UC Davis about metabolomics. Take it away, Camille.
Thank you so much. Yes, I'm going to talk a bit about metabolomics. and peripheral and central biomarkers of Alzheimer's disease. My name is Kamil Borkowski, and I'm associated with West Coast Metabolomics Center at UC Davis. Just to mention that this data that I'm going to show you today was generated and interpreted together with Dr. John Newman at United States Department of Agriculture in Davis, and also that this entire project is part of Alzheimer's Disease Metabolomics Consortium and part of the FNIH-funded Integrated Biomarkers Project, including collaborations with Emory, Columbia, and Duke University. And so we work mainly with targeted metabolomics of lipid mediators. Our platform uses high throughput methods. For this project I'm going to show you today, we quantified 145 compounds from a single extraction of 50 microliters of plasma, or 200 microliters of CSF. Our main focus are oxylipins and endocannabinoids, but we also quantify a handful of bile acids and steroids as a part also of a lipid mediators group. I would also like to discuss briefly why we use targeted metabolomics instead of untargeted approaches for biomarker discovery, because that's actually a question that we've been asked by a reviewer on our recent paper. I thought it's actually a really good and interesting question. So first, targeted methods have much greater limits of detections due to the noise reduction. So we can actually measure metabolites at the picomolar levels. And many of those lipid mediators are indeed at those kind of levels in biological fluids. Using targeted methods, you can, of course, select what you want to measure, and you know exactly, I mean, you can kind of speculate what you're exactly going to see in your biological fluids. Unlike with untargeted methods. And the results that we get are quantitative and very reproducible. And basically, we can compare the results between different cohorts. Couple words about oxylipins and the cannabinoids. Those compounds are derived mainly from polyunsaturated fatty acids. What you see over here is a metabolic map and actual coverage of what we measure in the plasma samples. So we have fatty acids in the middle, like for example, omega-6, omega-3 pathways, and lipid mediators being enzymatically and non-enzymatically generated from those polyunsaturated and saturated fatty acids. So basically by focusing on oxylipins and endocannabinoids, we cover multiple enzymatic and autooxidative pathways, both for omega-6 and omega-3 fatty acids. Now, there's a very good reason why we're focusing on those compounds, because those compounds are involved in almost every biological aspect. So they are key regulators of inflammation, energy metabolism, vascular function, neuronal function, and many, many more. So we are really getting a good screenshot of multiple aspects of metabolism over here. And also those compounds were reported to be involved in Alzheimer's disease pathology. Okay, this is a coverage of lipid mediators detected in plasma and in CSF. Those are the data generated from Emory cohort, where we profile approximately 300 plasma and matching CSF samples from Alzheimer's disease patients and healthy controls. So you can see we have much more going on in plasma than in CSF, yet still CSF has decent amount of lipid mediators. Now, I would like to talk a bit about the relation between peripheral and central lipid mediators. So those are Spearman correlations between plasma and mass CSF lipid mediators. Highly significant correlations you see over here highlighted in red. So just to summarize, we see strong associations between CSF and plasma bile acids and steroids, and only long-chain omega-3 polyunsaturated fatty acids and their ethanolamides. And we see no associations between plasma and CSF other polyunsaturated fatty acids, and now associations between oxylipins. So this lack of associations between plasma and CSF oxylipins basically shows the importance of lipidomic analysis in both plasma and central nervous system, as those two pools of lipid mediators, specifically oxylipids, most likely represent different regulation, and they hold probably different information. Now, let me make a brief summary of the key findings from this cohort. We found peripheral and central changes into the specific pathways, cytochrome P450, soluble epoxy hydrolase, abbreviated CYP, SEH. This pathway regulates inflammation and vascular tone. And fatty acid ethanolamides, which is a class of endocannabinoids, they regulate inflammation. They're neuroprotective. They directly activate CB1 and CB2 and TRPV1 receptors. Very important that metabolites from those two classes from those two classes can predict Alzheimer's disease with the Roc curve of about 0.9. And we are talking about both plasma and CSF metabolites. So basically we have peripheral metabolites that can be very informative of Alzheimer's disease pathology. And right bottom corner of the slide is a brief illustrations of those two pathways. So in CYP-SEH pathway, polyunsaturated fatty acids are being converted by CYP into anti-inflammatory and vasodilating epoxy fatty acid that are further converted by SEH into pro-inflammatory and vasoconstructive diols. Now, in terms of endocannabinoids, as I mentioned, direct activators of CB1, CB2 receptor, they can be removed and metabolized from the system by fatty acid amide hydrolase. Now, importantly, those two pathways can be targeted by non-pharmacological intervention. CYP-SEH pathway may be targeted by a SEH inhibitor, which can change basically the balance between the pro and anti-inflammatory mediators. Ethanolamides can be targeted either by inhibitors of metabolizing enzymes or by administration of exogenous cannabinoids. Just to mention, we found the same exact pathway, CYP-SEH being associated with cognition in a completely separate cohort of cognitively normal elderly subjects, which actually providing a further evidence for involvement of this pathway in the development of Alzheimer's disease. Now, I would like to briefly focus on the compatibility of metabolomics with other platforms, especially proteomics. So you see actually part of the data that Lenora presented before. And basically what we see that integration of metabolomics and proteomics data actually shows new connections that we can observe using, for example, gene co-expression network. So here on the left is an example of a strong correlations between plasma metabolites, especially CYP-SEH and metabolites, and CSF proteins affected in Alzheimer's disease, especially those involved in glycolysis and cell proliferation. And when you look on the right, on the gene co-expression network level, enzymes involved in metabolism of those lipid mediators do not co-express with those CSF proteins. So basically, integration of lipidomics and proteomics can bring new information about Alzheimer's disease. As for example, we see over here, vascular and inflammation regulators are associated with glycolysis. I think I did good on time. So basically, in conclusion, in our study, we identified both central and peripheral changes in two very specific pathways, CYP, SEH, and ethanolamines, which are associated with Alzheimer's disease. CYP-SEH pathway is also associated with cognition, which kind of give us a hint that this pathway may be involved in the development of Alzheimer's disease. Very important aspect is that peripheral lipid mediators can be informative about Alzheimer's disease. And also lipidomics and proteomics are complementary, providing additional information not visible on, for example, gene co-expression level. And thank you so much. I would like to acknowledge all our collaborators, members of the Alzheimer's Disease Metabolomics Consortium, which is funded by Foundation NIH. And thank you for inviting me. Thank you.
Thanks, everyone. Those were all fantastic, amazing talks. Obviously, we don't have any time, but I see you guys answered almost all the questions. I think it's time to go. I will say I really enjoyed all of this, except for the stress of having to manage the time.